Hello.
All right. Good afternoon, everyone. Welcome to the fourth Hub Cures speaker series. As you know, have been holding these speaker series every Tuesday at this time, and the goal is really to highlight the pandemic response from Massachusetts institutions, our academic institutions in particular, and and it sort of is fitting into the theme of our annual conference called MALSI, which will be virtual this year on August 20th. The theme of MALSI this year is collaboration fuels innovation. And there's nothing like the kind of collaboration that is happening in Massachusetts around our response to the pandemic. And frankly, with a little bit of hyperbole, I like to say that Massachusetts is going to lead the world out of this pandemic with all this collaborative activity that's going on. And there is no better institution that reflects this idea of collaboration that's so unique to the Massachusetts innovation ecosystem than the Wyss Institute at Harvard. And I'd love to welcome Don Ingber, who's the founding faculty member of the Wyss. And I would love to start off by Don, just asking you first to tell us a little bit about yourself and your background, and then we'll talk a little bit about the Wyss Institute.
Myself, I'm usually starting around the V, so that's a good taking me back. I've been a Harvard faculty member for 36 years. Also at Boston Children's Hospital. I'm the Judah Folkman professor at Harvard Medical School and professor of bioengineering at the School of Engineering and Applied Sciences. My work has spanned many different areas, so that I'm a trained cell biologist, MD, Ph.D., but I got early on very interested in mechanical forces in biology and got into engineering, computer science, nanotechnology, translational medicine. I guess I learned to collaborate early, and I learned about the power of the Boston-Cambridge ecosystem. So, I taken advantage of it in many different ways. I guess in some ways the Wyss Institute is sort of a culmination of that, all the things I learned. What I always said that many of the faculty I pulled together to form the Institute, we all had done innovative stuff. Many of us had collaborated, but we did it in spite of the academic models and systems, and that the Institute was really structured to enable and empower things that we thought really could transform how academia works and how it crosses over with industry.
Yeah, no question. One of the early, I think, collaborators in the Wyss was Jim Collins, who was my best customer when I ran the tech transfer office at BU. But also Judah Folkman, one of Judah's other proteges, Bob Langer, was our first speaker of the speaker series. So it shows you how everything's very interconnected in Boston.
It really is.
That brings us to the Wyss, which, by the way, I have to say that its byline, which is this idea of being biologically inspired. And I remember I've come to your events most every year, and you have architects from MIT, and you have all kinds of interesting people who are using biological inspiration to come up with great ideas. But I don't want to steal your thunder. Tell us a little bit about how the Wyss came about.
God, it's almost 15 years ago. I was asked to co-chair a committee with Dave Mooney to envision the future of bioengineering across Harvard and the hospitals by the provost. And long story short, we kind of looked backwards and we saw that engineering had transformed medicine over the past 50 years, and also non-medical areas as well, by taking engineering principles and applying them to particularly medical problems. Well, we saw that the boundaries between all the disciplines were breaking down. We were all collaborating with each other in physics, computer science, material science, medicine, biology. And we realized that most of the funding agencies like NIH focus on, how little we know the reality was that we learned a huge amount about how nature builds, controls, and manufactures down to the nanoscale. And so we felt that we could flip that vision of bioengineering on its head by basically focusing on leveraging how nature builds and manufactures and controls. So essentially leveraging biological principles to develop new engineering innovations. And that's what we called at the time biologically inspired engineering. You know, I have never taken an engineering class in my life yet. I do this. George Church is a geneticist. George Whiteside's a chemist. Jim Collins was trained in control theory. You know, yet we all have been doing this without even realizing it. And so we kind of pulled these people together, brought in young people, and just made it really happen.
That's awesome. And so it went from this committee and sort of looking at how that kind of collaboration and, you know, with that biological inspiration at its core, how did you get it funded?
We very lucky to come across very early on a donor, potential donor philanthropist, Hans-Jörg Wyss, who had come to Harvard and been a little taken aback by the way it was siloed in terms of the different schools not talking, different faculty not talking. And so we put this vision together, and it was really, Dave and I reached out we said we used a lot of shoe leather, and that we reached out to faculty across not only all of Harvard, but the hospitals, MIT, BU, Tufts, et cetera. And we gelled, everyone loved this vision of biologically inspired engineering. But the other side of it that was there from the beginning was that this wasn't going to be just a research institute, but really a translational institute. That we really believed, as we say on the website, that, you know, breakthrough discoveries don't change the world unless they leave the lab. And many of us had done entrepreneurial things, and we really felt that there had to be a new model. So from the very beginning, the vision was one of bringing in people with industrial experience, not to run cores, but to be part of teams and to come up with new structures for collaboration, not giving faculty lab space in individual faculty labs, but giving program areas. And we got the donor, he got very excited about this idea. And, you know, long story short, in 2009, he gave what was at the time the largest single gift in Harvard's history of $125 million. And when you hear about big gifts, it's usually for a building. This is all for operating funds. And so that really allowed us to bring full force to bear and really make things happen.
Yeah, you know, I like to say that, you know, the secret sauce of the Boston innovation ecosystem has two sort of core elements. Number one is what I call geographical serendipity, which is it was never planned that, you know, Harvard and MIT and Northeastern and BU and partners and, you know, Brigham and all these would be within a 3-mile radius of each other. So it just ended up that way. And there's just no other city in the world that has this density of innovation. And the second, of course, is how good our academic institutions are at philanthropy, and particularly around translational philanthropy. The Broad Institute, the Wyss Institute, Blavatnik, and all this, everybody, Tufts, everybody's been good at it. And so there
was another bit of serendipity, I should say, which was 2009 is when the stock market crashed.
Right.
And we were supposed to all be put into a building into Alston. But because of that, we actually were able to rent space in the middle of the Longwood medical area, where we are arm's length from all the major hospitals, you know, almost all. And so, you know, from the beginning, we planned to be a consortium where partner institutions would be all the Harvard hospitals, which they are, but also MIT, BU, Tufts. And so we structured it to harness the Boston, Cambridge region, exactly what you said. So we have partnership agreements to let faculty, students, and staff be on site at our institution. And the overhead comes there. The IP is owned by Harvard and the Institute, but there's already royalty distribution agreements in place. And so people can move and flow and just collaborate and not really worry about, you know, dealing with those sorts of issues that often restrict how people interact, even across the Harvard area. I mean, the Boston, Cambridge area.
Yeah, that's fantastic. And of course, the Wyss has spun off many companies.
Yeah.
In some ways, it's becoming, I akin it to the MIT Media Lab. So the MIT Media Lab was this very prolific place that was spinning out companies from MIT, and the Wyss has sort of become, the prolific place that's spinning things out from,
Yep.
Harvard in particular. So companies such as Emulate, any others that come to mind?
Sherlock, which just got approval for the COVID-19
We
test.
just had Raul Dunda, the CEO, on as a speaker.
Yeah, yeah. So yeah, there are, I think, about 40 companies that we've founded. We're a different model than the Media Lab, and that the Media Lab, and I know this because the leaders of it came to visit our place to figure out how to change. They didn't really have ownership, so faculty would develop ideas, and then when they looked like they were interesting, they would take them out of the Media Lab and then do them separate from the Media Lab. So we really try to nurture entrepreneurs and mentor entrepreneurs, young ones at the Institute, and basically de-risk technologies so that when it goes out as a more mature company and more likely to be successful.
Yeah, no, that's a very good model. That's look, I'm an engineer in the tech world who came into BU to teach, and then ran the tech transfer office, and we launched 8 venture-backed companies out of BU while I was there that have raised over 350 million to date. But the one thing I realized is the longer you can keep them in the academic environment, the better.
Yeah.
Because
there's versatility. You can bring new people in and new thoughts, ideas. You have access to the capital equipment of our. We have 375 staff, and we could access equipment at any of our collaborating institutions. So you can't do that in a small startup where it's focus. It really does give greater strength.
Yeah, and of course all the conflict of interest rules and things like that. So they all start kicking in once you actually cut the cord and head away from academia. So the longer you can keep it there, the better. And you have access to non-dilutive funding, you have access to the course, all those things. So things were going along at the Wyss, and then the pandemic hit.
Yes.
And what did you think? And when did it start occurring to you that maybe you should pivot the resources of the Wyss towards a response to the pandemic?
Well, first off, the Institute, the whole model and the first gift was contingent upon us creating an innovation organizational structure that could have near-term impact. So this is very different than getting a paper and then someone a few years later hearing about it or a patent and a company license. It was doing it quickly. So we are set up to really do things fast and to focus on problem backwards. So when COVID-19 happened, there were few of us that I think turned really quickly. I mean, I had been personally funded by DARPA on a program for 2 years already called Prepare, which was literally to be prepared to develop therapeutics for influenza pandemics that we knew are going to emerge in the future. I had developed technologies called organs on chips, which are microfluidic culture devices that are lined by human cells and tissues that have vascular flow, and in the lung you have air spaces and breathing motions. And so we were working on influenza therapeutics. When the paper came out in mid-January, something like January 12th, on the SARS-CoV-2 gene sequence, the day after, my team, and this is bottom up, my people, basically engineered a CoV-2 spike protein pseudovirus. So I would say that my team was working on it from January 13th. I am sure that other members individually were working on things early and beginning to pivot. People like David Walt, who very quickly got diagnostics up and rolling. Jim Collins had been working on RNA-based therapeutics for dengue virus, and quickly started to pivot. It was the beginning of March when Harvard began to reach out and give us a heads up that we probably were going to do a phase down of operations, and that only those who would continue COVID-19 related work could stay on site. Now, most labs shut down completely, but people had to write proposals, and the director of the institute, which happened to be me, had to review all these and basically decide whether they really were COVID-19 related and give approvals. Within one day after closing down, we had 38 people working on site. So this was from about 5 to 6 different lab groups. And our lab groups are faculty focused groups, but they also have staff, but we have about 40 staff, I mentioned, with many years of experience from industry, and they cross faculty labs and cross the institute. And we had a mixture of technicians, students, fellows, et cetera. And so we were working from mid-March specifically on COVID-19 in a concerted way. And we were working on, personalized, equipment, safety equipment, N95 masks, face shields, nasopharyngeal swabs for diagnostics, as well as vaccines, diagnostics themselves, and therapeutics. So the complete gamut.
That's interesting. How come you guys were working on the nasal swabs or PPE, that category?
I got a call from Rami Arnaud, who's the co-director of the clinical lab at the Beth Israel Deaconess Medical Center. He knows the Wyss, and this is, again, beginning in March. The clinicians, I got various groups reached out. The clinicians saw the wave that was coming. They had enough nasopharyngeal swabs, but they knew they wouldn't last. And so they desperately were reaching out to people. This was true for the masks and for face shields, for all of the areas where it was clear immediately they were going to go quickly. So we had never worked on anything related to nasopharyngeal swabs, but I got one of my staff members, Richard Novak, who we do a lot in manufacturing, 3D printing. Richard is involved both on therapeutics projects, but also on a project that is doing a microfluidic system that has engineered parts, molded parts, 3D printed parts for water purification. So I pulled him on and said, would you get involved? I pulled some other people on to work on, who are material scientists, to work on the project as well. We started with Rami sending us pictures and samples of what they really look like, and real ones, which are these thin, narrow applicator stick type things that have some flexibility, and they have a little flocking on the end that, if you go under a microscope, looks almost like toothbrush bristles that are randomly organized to collect mucus. We started by thinking we would 3D print the handle, and then we put a little coating on it. And in the water purification project, we collaborate with the company 3M. We do a lot of, besides startups, we collaborate with lots of companies. And so I wrote them, and they immediately, within a minute, wrote back. Within a day, I had 8 people there looking through their inventory of existing material, and they sent it to us the next day, and we started testing them. That's just an example. The way that scientists, engineers, companies have collaborated on this has been inspiring. Right. If only the government could do 1/zillionth of it. So it turned out that then Rami and others had reached out to many different people. Another faculty member at the Wyss, Kit Parker, who's also at School of Engineering, he got involved with 3D printing companies, and they started to work on the nasopharyngeal swabs totally independently, but also connecting with Rami. They sent materials to Rami. By the way, everyone, including the companies, were putting designs out in a STL format, a file format. You could put it up on the web, GitHubs, everybody could share, everybody was trying. And they made great headway on that. And we were making good headway, but we realized that there may be a simpler way to do it without the flocking, given that 3D printing didn't have flocking. So we went to an injection molding, because we had worked with lots of injection molding companies around many projects, and Richard knew of them. So we got prototypes made that had a simpler design, because 3D printing is fantastic, except it's like 5 companies that could do it, and they're expensive. But they very quickly, I think by April, could do like 1 million a week, and you could buy them. by the way, they were tested at the Beth Israel for ours and theirs for PCR compatibility. They all passed, and then they moved on to mucus collection and did some preliminary clinical testing. But we moved to an injection molded version because we could see that the supply lines were really dropping. This is when it was a craze of desperation out there, and that there are injection molding companies anywhere in the world. So this could be useful in South America, in Africa, and so forth. And very quickly we got something that looks like a honey dipping stick that when you put into honey, and tested it, turned out to be more comfortable for patients because it didn't have all these little bristles.
Yeah.
It had similar PCR capabilities, and that has now been licensed to 2 companies and about to be 3, and they're manufactured, again, millions a week, but they're half the price of the existing ones.
Wow.
Which is amazing. Now Harvard and I think Stanford and many institutions have agreed to give royalty-free, non-exclusive IP, licenses out, and so I think it's great
for a period of time, not forever.
For the first year, and then it gets re-examined in the future. Absolutely.
Right.
And so the nice thing is that we have multiple companies in the United States, we have in Canada, and there's some for South America already. So that was something where it's not like we were doing that, but that we had the capability to just pivot and use know-how. There were people I had staff who have 3D printers at home that they were doing some of this. There were other people that we brought in that had worked with the FDA before, so they reached out to FDA to get some regulatory input on it. Others were researching every manufacturer of injection molded products who can do can get a FDA approval can sell products and sterilize. There are not many, amazingly. So it was kind of, we had staff people who are not graduate students, they're not postdocs, they've been in industry, they can see both sides, and they just want to solve problems. I think that's really the key.
Yeah, and it's interesting your point about government. I mean, certainly the federal government response has been dismal at best. But I think the Massachusetts government has responded. We had Carolyn Kirk speaking. She was running this, the Manufacturing Emergency Response Team, the MERT. They put out $45 million. They got 27 manufacturers in Massachusetts to pivot to making PPE, and they've had 4 million masks and other things. You guys weren't involved in that, were you?
We were involved early on in various meetings. There were groups of manufacturing meetings, but not directly in a serious way.
So that's kind of one PPE story. Do you have any others, or was it small?
Yeah, I mean, Jennifer Lewis, one of our faculty member who is an expert in sort of digital fabrication, known for 3D printing, and one of our staff who's really an expert in that area, and very creative, James Weaver. They got involved with face shields. People reached out to them. They ended up not doing 3D printing as much as using simple, laser cutting, and very inexpensively. And they delivered over 1,000 of those by hand to Boston Medical Center and I think St. Vincent's Hospital in Worcester, which are less wealthy hospitals that really desperately needed them. They've worked on N95 mask replacements. They work with people at Children's Hospital on ergonomic masks, on intubation shields. So when they're intubated, there's not a lot of vaporization of. So yeah, I think and I should say we also, David Waltz, one of our faculty, was the co-head of the partners, the Mass General Brigham Partners COVID-19 Innovation Center, and they were trying to organize things for partners provisions. And David was a co-leader. He connected me with Gary Tierney, who I knew before. And as a result of that, they saw what we were doing. And so Richard Novak, working on the nasal swab, became the leader of that working group, and Jennifer and James on the face shields groups. I have other people in diagnostics, Paul and Jolie on a diagnostics group. So everybody just wanted to help. So we got involved in many different groups, and these are our partner institutions. So having that kind of connection, and we feel a responsibility to help them. And I think they were appreciative that we had capabilities they didn't have on site.
Yeah, that's such an awesome story. Let me switch gears a little bit and let's talk diagnostics because, testing, we hear this around the world that the way you contain this pandemic is test, trace, and isolate.
No.
And the tracing part, Massachusetts has put up a contact tracing group. I'm involved in a MIT media spin out that's doing digital contact tracing. Very similar experience. We set up a nonprofit. We raised $2.5 million. We had 1,000 volunteers working on it. It's open source software. It's available to everyone, and it's moving really fast. But testing has been a bottleneck, and obviously the standard of practice is qRT-PCR, but there's all these new technologies happening. I understand that there's several coming out of the Wyss, including lamp-based technologies and others. Can you tell us a little about the diagnostic side of things?
Yeah, as I said, David Walt, is the founder of Illumina and Quanterix, and he's on when the National Academy formed a committee early on this around COVID-19 around emerging threats, he was on it. So he's been working on it, and he has a lab at the Brigham with with CLIA lab capabilities. So they've been working with clinical samples early on from I think February time, and so he has been working on serological testing using his Simoa, which is single molecule detection. And they have a Med Archives paper where they got this early on, 86% sensitivity, 100% specificity, or I may get that backwards, for the first week after infection.
Wow.
And then 100% for both thereafter. And the Massachusetts the Commonwealth population-wide study chose 2 assays to use, and that was one. So that was one place where we were involved with he was involved with the state. He's also now moving on to saliva-based testing. Peng Yin has developed DNA nanotechnologies that give you very high specificity, sensitivity nano switches that can give colorimetric readout, for example. And he has been, and Jim Collins has also worked on, DNA switches as well. And they both are working on sort of LFA-based tests, meaning lateral flow assay, pregnancy type tests, right? They give you the line. Now, there are many companies doing this, but their sensitivity is extremely high. So that was something they started on way early on. My team, and collaborating with Jim, actually, we had developed just this last year electrochemical sensor technology. And electrochemical sensors are what's in the glucometer, that diabetics use, right? So they're simple, cheap, and quantitative.
Right.
But they're usually enzyme-based, because if you put antibodies on them, there's a high non-specific binding and background. We came up with a nano-structured coating, very simple, that gets around that. And we can actually get incredibly high sensitivity and specificity, and you can pack them in, multiplex in a small area and get quantitative readouts. So we've been working on, multiple analytes, including IgM, IgA, IgG, potentially viral antigens as well, in a electrochemical sensor format. And Jim had developed paper-based diagnostics, which he had done for Zika very inexpensively. Right. So we're kind of collaborating with similar sensing, molecular assays, but doing it in both paper-based on electrochemical. George Church and David are collaborating on next-gen sequencing with the possibility, they think, of doing and analyzing 10,000 samples in a single run. So there's, and then Sherlock partly came out of the Wyss, and they have the CRISPR-based diagnostics that got approved. There are others, but that I can't even remember right now. But yeah, the full range.
Yeah, I know, it's quite amazing. And next week we're going to have some speakers talking about the RADx program that NIH has allocated $500 million, which is being managed right here in Massachusetts by CIMMYT, with UMass Lowell and M2D2 being one of the centers, Emory being the other. And they, last I heard, they had received over 2,000 proposals.
That's great.
And so it's an interesting horse race, but we're talking about standing up millions of tests a day by the fall. Do you think that's achievable?
I mean, these ideas, these creative ideas like the next gen sequencing raise that possibility. I mean, I was on calls early on with economists who had pressure from, who had discussions with people at the White House that they wanted to get tests right away, and they wanted to basically take 2,000 patient samples and mix them together and do that, dilute them together. And I reached out to all the clinicians that I could and to all the experts, and it was a resounding no, do not do this. And I'm glad that they did not do that at the time. But I think clearly the need is there. But I think, the answer is going to be a combination of testing that is possible, plus therapeutics that we can get out there, plus vaccines if we can get them to work. The one thing I want to add, I forgot about diagnostics that's creative, is Jim Collins had been translating his paper-based diagnostics, which uses basically cellular extracts that basically doing synthetic biology rather than in bacterium. You break the bacterium open and you put the cell extract in paper, dry it, and it turns out you can rehydrate it and you get functional activity. And you put complex sensor readout assays in there. He's been working on it in fabrics, and with COVID-19 he pivoted and he's working on it in face masks.
Yeah, I know. I read
that's the kind of idea that you could basically have thousands of them and people could read out themselves that within 2 hours they went to a party or sang in a choir and all of a sudden their mask turned blue. Or in a nursing home, you can actually, identify there's a point of spread and watch it spread, and especially when there's limited healthcare workers. So I think those are kind of creative ideas coming out of the box, totally out of the box types of approach.
So the question is, with all this diversity of ideas and everything getting funded, how are we going to sort out the winners, or will the market sort it out somehow?
One thing that I've found wonderful about this, and there's a lot of Twitter discussion, is what we've gotten outside the journals and the review process.
Yeah.
Where people are just putting things up on bioarchives.
Right.
And basically, in 3 months, 2 to 3 months, you have people either confirm it or prove it wrong. I think the hydroxychloroquine is a great story, right? One paper comes out really quickly on a few patients. It goes to the government, it gets expanded, it goes out of control. But within 2 months, there were already papers saying it does not work, many papers that does not work. Now, no one listened to that, but it was there. Now, if we had been non-COVID-19, it would take a year to a year and a half to get it published. It would take another year and a half for people to do repeat experiments and then get it published. And that has allowed the marketplace and scientific marketplace to test, prove, validate, or test, prove, disprove really quickly. I think this is the way things really have to be if you want to have impact.
Yeah.
And forget what impact is really.
Yeah, it'd be great if we sustained that. But yeah, you're right. I mean, there's, I just read about a paper co-authored Brigham that had to be retracted because the underlying data was suspect. But you're right. I mean, these cycles are happening really fast.
Normally things get in and then it takes years to bring it back out. Yeah. Where here it's being done in weeks to months. And it's sort of like organic growth. You grow and then you call back, and then you grow and then you call back. I do think that it's going to, there's a lack of coordination. I was just on a call yesterday and will be tomorrow on a webinar about the lack of coordination in clinical trials across the nation, across the world, is going to be a problem because each one's using different validation assays, scores, experimental design. Yeah. I think that's true with diagnostics, and I think that's will hold things back. But right now, things are moving so quickly that it's probably going to be the marketplace.
Yeah, I mean, I'll be honest with you. I mean, I've experienced this before as a tech entrepreneur. It was called the dot-com boom. But the thing that led to the dot-com boom was open source software. And that whole open source software movement was started in Cambridge, actually, right across this, on Broadway. That's where first open source software foundation was. It was famously, stated by Bill Gates that this was socialism. But as it turns out, it wasn't socialism. It was just a new business model where it was crowdsourced, software development, and it became very effective. It started forming the basis for the internet. Linux forms the basis for large portions of the internet. So lots of this open source software. In a way, this is almost like that, but biology, right? So it's almost open biology, and it's very interesting. So let me pivot a bit towards vaccines and therapeutics. I assume Wyss is involved in both those areas as well?
Yeah. So the vaccines, David Mooney had developed a biomaterials-based vaccine for cancer that already went to phase 1 clinical trials and did well enough that Novartis licensed it about a year or so ago. And so over the past year or two, his group and partly my group had been using it to develop broad spectrum infection vaccines, largely bacterial, and it had worked incredibly well, actually. And so they pivoted when this happened, and they've been using commercially available, inactivated, SARS-CoV-2 virus, did mouse studies, got tremendous results, high titers, IgG, neutralizing antibodies, and are now actually starting animal studies with needle at BU. So that's the only vaccine work we have going. My team has a major effort. We just got a $16 million DARPA grant on repurposing FDA-approved drugs for COVID-19. But I mentioned before that Jim has worked on RNA therapeutics where he found good activity against dengue. He's starting to work on COVID-19. I've been working on DARPA Prepare with RNA-based therapeutics against influenza. We now have good data that they're very potent against influenza, SARS-CoV-2, and other coronaviruses. You might have broad spectrum therapeutic for future emerging threats. But the DARPA program around COVID, around repurposing, I think, started because we had pivoted, I mentioned in January. We made pseudotype virus. My team, some of them were virologists, and so we got the cell lines that virologists screen in, and we tested drugs that had worked against related SARS viruses and MERS and Ebola right off the bat. And we got nice activity in this assay using a pseudotype virus with the specific spike protein from CoV-2. Because we use these organs on chips, and we know that cells in a dish are not the most relevant, plus the cells that virologists use don't have high levels of the natural receptor ACE2 or the enzyme TMPRSS2, and often have suppressed interferon systems, which is why they get good replication. So we tested the same drugs in our organs on chips, human lung airway. And the nice thing about organs on chips is we can give the drug at a clinically relevant dose and exposure under flow at a Cmax we did. And there we found only a subset, only a couple of the 7 drugs that worked on the cell lines worked in the chips. And that got DARPA's attention. They knew we were doing this. They had reached out to me. I've also been funded by Gates, and they had reached out to me. Between the two of them, they connected me to groups that have BSL-3 labs and are working with the native virus because we only have a BSL-2, which is why we work with pseudotype. And very quickly, we were asked to submit an application to develop a systematic way to deal with drug repurposing because the hydroxychloroquine was so clearly out of control and other things like it. And everybody were publishing results with these cell lines, and everybody's wanting to start clinical trials. So we developed a pipeline where basically we are testing it in those cell lines. We are then using these organs on chips where we can not only look at infection and replication, but we can look at inflammatory response, cytokine response, immune cell response. We've done all this with influenza on chips. It's absolutely beautiful mimicry of human clinical responses. And we're actually getting the organ chip instruments that are now commercialized by Emulate into the BSL-3 labs. And we're doing parallel work now with related coronaviruses. And then we have an animal model at another collaborator lab, hamster model that looks very nice in terms of viral infection. And we're linked with discussions with groups in Boston, again, at MGH and Brigham, the groups leading clinical trials at those institutions. And I was amazed when I talked to them because I thought they'd have no interest in what we're doing. And it was like, my God, this is the first time we're hearing about maybe having clinical trial proposals where there's data that it works in some way against the COVID-2 virus, because everything else is, it works against interleukin 6, so let's try it, or let's try this, or let's try that. So we are trying to develop a expedited pipeline to go from cell activity to a human organ type model, highly differentiated, high ACE2 expression, TMPRSS2 expression, have some animal data if necessary, and to get it into sort of an adaptive clinical trials design, which can be very rapid. So we have, there's a lot of therapeutic stuff going on. Other people, George Church is working on antibodies against the spike protein in a sort of high throughput sort of way. There are many projects going on, but I think those are the big emphasis right now.
That's awesome. I mean, it's just amazing to hear all this activity. And, of course, I don't think this is going to be the last pandemic in our lives. I mean, it's been 100 years since a major one, but, with the world urbanizing the way it has and, displacing wildlife everywhere, I expect we're going to see more of these, because I think SARS-CoV-1 was, 15 years ago? A little over, something like that. The countries that got hit with it, the one to the north of us, for example, Canada got hit with SARS-CoV-1. We didn't. They were much more prepared for SARS-CoV-2.
In Asia, yeah, exactly.
Asia, of course, was much more prepared because of that in H1N1. But it's clear that all this work that's going on is not just for COVID-19 and SARS-CoV-2. I think this is going to be needed for the world. But we thank you so much for taking the time. I hope you'll stick around for a few minutes. We've got a bunch of people, I'm sure, who'd love to talk to you. And I'll ask Priya to pull us back onto a table, and people can start networking now on the tables. So thank you.
Thank you. Pleasure.